Hello everyone, we are going to start with Objective 7 during this flipped lesson kind of podcast. And Objective 7 says compare and contrast DNA and RNA. And you have the spot in your notes where you have these two titles, DNA and RNA. And I don't want you to necessarily write anything down on this slide, but when we go to the next one, I want you to kind of list those differences just as bullet points. DNA contains the code to make the protein. However, the code is the wrong language. Ribosomes can only use RNA to make protein. And so we talked a little bit about RNA making protein yesterday. I kind of talked about how RNA is like the ticket that the waiter might give to the cook to make whatever protein or whatever food it is. So DNA has to be turned into RNA, and we went over this process called transcription, real brief at the end of the hour yesterday. So what's the difference? Give me 1 second here, and I need to make my slide a little bigger. So here you go. Basic differences here is that DNA is double-stranded versus RNA is single-stranded. RNA also is not a helical shape. It doesn't have that spiral shape. The other big difference is DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. And you can see what's underlined on each of those second bullets there. Deoxyribose versus ribose sugars. And remember that OSE right here, that means that both of these are carbon sugars, and it basically just has to do with their chemical structure. It's not that big of a deal, it's just how we name it. And the other big difference, and the one that you really need to kind of focus on as we go through this protein making process, is that RNA is substituted with uracil as opposed to thymine in DNA. Okay, so all of the T's are U's in RNA. So if you can kind of see here, this is just some of those major differences here. What I'm going to do here with this laser pointer is I'm going to try and show you why this is called deoxy. So you've got your phosphate group right here, you've got a 5-carbon sugar, and you've got a nitrogen base. This could be an A, T, C, or a G right here. And the reason why we call this deoxy is because there is one less oxygen right here on this second carbon. You see that it's just bonded to this hydrogen right here. But if you look at RNA and look at a ribose sugar over here, we see that there's this OH right here. Or in other words, there is an oxygen right there. So we call this an oxygenated sugar. Or better way to say it is this is a deoxy sugar. It's deoxygenated. Other differences, you have T versus U. The other three nitrogen bases stay the same. And then down here you can also see that you have a double-stranded helical DNA crystal structure versus in RNA. It is just this single-stranded with, it almost looks like half the ladder. You have the phosphate, you have a ribose sugar, and then your 4 nitrogen bases. Alright, that kind of does it for Objective 7. Just real brief, we were going to explain the difference between DNA and RNA. Objective 8 says explain the 3 types of RNA and how they aid in transcription and translation. And we haven't got to translation yet, but we have got to transcription. We talked about that just real brief. mRNA is the one that we're going to spend the most time on because that M stands for messenger RNA, and it's what it carries the copies of the DNA code into the cytoplasm to this organelle that we haven't talked about a whole lot, but maybe you've recognized the name, something called a ribosome. And ribosomes are where proteins get made, they get pumped out. rRNA, stands for ribosomal RNA, and this RNA simply makes up ribosomes. It makes up the parts of the ribosome. Ribosomes are made out of RNA, and the specific kind of RNA that they are is ribosomal RNA, which is what this small r stands for. And finally, tRNA, this is called transfer. That T stands for transfer RNA. And we'll learn about this a little bit more when we get to translation. But these tRNAs what they do is they will carry what we call amino acids to the ribosome and link them together. And a protein, the end product, is just a chain of a bunch of links that we call amino acids. So you think of a chain, and you think of all the links that are there. All those links are individual amino acids. We sometimes call it a polypeptide chain as well. But amino acids make up proteins. And each one of those amino acids is carried to a ribosome by a tRNA. Okay, transcription. Explain the process of transcription. Number 9 is a little bit more of a lengthy objective. So the process of turning the code of DNA into RNA and sending it to the ribosome. By the way, the ribosome is not inside the nucleus. And so you will see the DNA change into RNA, but then that RNA piece has to leave the nucleus and go to a ribosome that's located just outside in the cell somewhere, what we call the cytoplasm. And this is known as transcription. Process works very similar to DNA replication. Your DNA is going to unwind and unzip so that we can get at it. Kind of similar to DNA replication, you have another enzyme called RNA polymerase, which actually matches nucleotide bases to the RNA strand by using the DNA strand as a template. The only thing that you need to remember here is that the T's are not T's anymore when RNA is made. They are replaced with uracil or U's. And so what you have here after that happens, and what we ended up with yesterday, is mRNA is produced as a single strand. So what happens after mRNA is made? 3 things happen. This is called RNA processing. When RNA is completely done being made, what happens is it gets some tags, basically, to tell the cell that it's done, that it's ready to be made into protein. One thing that it gets is a cap. Okay, a cap is just that it's actually called a carboxylated cap, but it's just something for protection so that the cell doesn't see that it's kind of this foreign object and want to destroy it. If it doesn't get this, if it doesn't get a cap, if it doesn't get this poly A tail, A again is just for adenines, but it'll get this kind of string of adenines at the end of it. If it doesn't get these 2 things, then the cell doesn't recognize it as being complete, and what will happen is the cell will try and fight against that piece of RNA and usually will destroy it. Okay, so both of these things right here are used for protection. Now the other thing is here, I'm going to try and draw this at the bottom, there's splicing that needs to happen. So let me grab my pen here real quick, and I'll see if I can, with my red marker here, yeah, okay? You have this piece of RNA, and this piece of RNA has pieces to it. I'm going to kind of draw it like this. And some of these pieces are used, and some of them are not, or some of them are coding regions and non-coding regions. So maybe you have an I here, that's a poor I, excuse me, and an E, and maybe another E, and then an I at the end. Okay? Well, this whole thing is not needed. All that we need in order to make proteins are the sections that we call exons. And I know that sounds a little bit weird because exons stay in and introns stay out. And so in other words, introns are sections of mRNA that are removed, and exons are the pieces that are left behind. So what's going to happen at the very end here, this splicing procedure, is basically you're going to have this I, or this intron part of the RNA, be cut out. And this I over here is also going to be cut out. And the only thing that you're going to be left with, your mature RNA, your mature mRNA, is going to be this piece right here. And so you're just ending up with the exons that are going to code for a protein and go to the ribosome. Another thing that you can kind of see here is there's something that we call, this is a better diagram than what I drew, honestly, but if you notice right here in blue, there is this, there's one exon. This whole thing is actually considered a gene. And so you have 3 different exons, and before it can be made into a proton, notice here that you have intron 1, intron 2, as well as exons 1, 2, and 3. And before it can be made into a protein, these introns right here get cut and they get thrown out, and then these pieces get glued back together so that right here all 3 exons are put in a linear order and can be sent to be made into a protein. Another thing you'll see here is this example of a piece of mRNA and its finished product, which you actually put on the last slide, is it's given this cap, 5' cap, and it's also given this poly A tail. Again, it needs both of these for protection. If it doesn't have those, then the cell's going to take a look at this guy in the middle here and it's going to chop it up, okay, and say, nope, that's no good. So, what happens when mRNA is complete? Excuse me. It goes to the ribosome, okay, and the ribosomes are located, if you want to write this down, let me see here if I can grab my pencil. These are located in the cytoplasm. Okay? Ribosomes are located in the cytoplasm, not in the nucleus. So this mRNA travels outside. That does it, because we are going to talk about translation in class today, and I hope you enjoyed this short little lesson. Hopefully it didn't take up too much of your study hall or your stickman golf time, but hopefully you can kind of look at this on your own and write these notes in on your own. And thanks for watching. Bye.